Hi, this is Mr. McLean, and in this video I want to take a look at the process of RNA processing or post-transcriptional modification that occurs with the eukaryotes. So in a previous video we went through the process of transcription, and we know that there are some definite differences between how it works in the prokaryotes versus in the eukaryotic cells. In the prokaryotes, they could actually manufacture messenger RNA sort of on the fly and then start translating it because there's no separation with a physical nucleus, and we also don't have the necessary modifications to take place. So when we start looking at a eukaryote, a couple of things that we want to emphasize is that there are really a couple different major changes. The first thing we want to focus in on is how the molecule is modified on the two ends. So we're going to see that on one side we're going to have the 5' end, and it's going to be modified with what's referred to as the 5' cap, and we're going to see that that is a guanine triphosphate. And then on the 3' end there's going to be what's referred to as a poly A tail. And we're going to notice that both of these modifications are going to be done to help facilitate the movement of the messenger RNA out of the nucleus. They're going to protect the messenger RNA from any of the hydrolytic enzymes, and they're going to assist in the binding to the ribosome. So when we take a look at this particular segment of code, this is going to be representative of some of the big modifications. So you can see over here on the left side we have our 5' end, and the 5' cap is the guanine triphosphate. So we have our 3 phosphates with a guanine on the end of it, and like I said, that provides us some protective functionality. When you see the 3-letter UTR, that stands for untranslated region. So it's a section of code that is not going to specify any specific proteins. So it's going to be classified as an untranslated region. And then we'll notice that we have our start codon right here. So our start codon is going to be AUG. This section through here has not yet been modified in this image, so we're going to focus on that a little bit more afterwards because there's some changes that happen internally, but you are going to have your segments of code that'll specify a specific protein. And then at the end, you're going to have one of the 3 different stop codons, which will end translation. So our translational unit is going to be really from here to here. At the end of that, we have another untranslated region. At this point, we call it the 3' UTR, and it is a polyadenylation signal. So the sequence of code that makes up a polyadenylation signal is exactly what we have right here. A, U, A. So once again, A, U, A. So that is another untranslated region. And then at the far end of the 3' section, we have what's referred to as our poly A tail, which is a very long sequence of adenines at the end of that. So when we start looking at some of the internal modifications that take place, we have what's referred to as an RNA splicing. And in order for us to get our messenger RNA finally put together, there are sections of code that are not coding for proteins. So the non-coding regions are the introns, also known as the intervening sequences. The regions that actually do specify code are called exons. Okay, and just think about it. Exons are expressed. Another way that I always keep these two straight is the modifications take place inside the nucleus. So if it is modified and it stays in the nucleus, it's an intron. It stays in the nucleus. It's not going to be able to be translated. If it's modified and it exits the nucleus, I would say it's an exon. So the exons eventually become the sequences that we will use to determine how the amino acids are put together. So the way in which we cut and join is known as RNA splicing. So if we look at this picture right here, we have a section of pre-messenger RNA, and we've already started to make some of the changes, the poly A cap or poly A tail and the 5' cap, but we notice that there are sections of exons and introns kind of interspaced throughout the overall sequence. So what we need to do is we need to extract and remove those intron regions so that we get a continuous sequence of exons. That's our coding segment. So how we do that is we use a structure known as a spliceosome, and spliceosomes are made up of proteins, and they are also made up of these small nuclear ribonuclear proteins, otherwise known as SNRPs. Okay, so what the SNRPs as part of this overall setup will do is we'll see a section of pre-messenger RNA. Here's an example of a SNRP along with some of the other proteins. When they collaborate together, they establish the spliceosomal unit. So the spliceosome is going to allow the SNRPs internally to find areas where there's complementary binding. So you're going to notice that these codes right here will complement the codes that are in that pre-messenger RNA. And we're going to see a little bit of that on the other side as well. So what it's going to end up doing is it's going to kind of loop this out and create a cut. And the area that's cut out is the intron. So that is how we're going to be able to remove the intron, and what remains are only exons. So, if you think about it, this is an enzyme that is not a protein. It's actually a ribozyme that's part of that. And, that kind of changed our thoughts on how enzymes had to exist, and the fact that a lot of these could actually be RNA based, or at least some of them. All right. One of the other things that was a major change, and this really became more evident after we finished sequencing the genome, is it became very, very obvious that we can make more proteins than what we actually have code for. So that brings about the idea of alternative splicing, alternative RNA splicing. And basically what it says is that in some of our more complex organisms, such as humans, our genomes don't have to be as massive as the number of proteins that we can make, because we can splice different things together in different ways. So an example of that is, let me get a little bit of space here. So if I were to say that here's my section of code, and I'll make these darker sections my introns. Okay, so if I have a section of code, so we'll just call this I1, I2, I3, we'll call this E1, E2, E3, E4. So I could potentially cut out this, this, and this, and make a sequence that goes E1, E2, E3, E4. But I could also maybe treat this as an intron as well. So I could have a different sequence which goes E1, E2, E4. So that's going to be different because we treated this as though it were an intron. So what that's going to do is that's going to allow me to actually have a completely different protein manufactured from the same original DNA. So that gives me tremendous diversity. And if you take a look at what these different exon sequences are actually coding for, is we say that they play a key role in developing the different structural domains of the overall protein. So when you think about our protein, it's going to be made up of multiple domains and multiple polypeptides. So hopefully that helps clarify some of the things in regards to some of the modifications that are going to take place. And like I said, the big three things to keep in mind is that whenever we finally put together a full messenger RNA molecule, you're going to have a 5' cap, you're going to have a poly A tail, you're going to have a sequence of only exons, the introns have been removed, you're going to have a 5' UTR right here, you're going to have a 3' UTR. The UTR over here is the polyadenylation sequence. You have your start codon, you have your stop codon. Okay, and that's a general layout for a eukaryotic messenger RNA. So in my final video, we'll take a look at the translation process.